Well, good afternoon and welcome to today's webinar looking at science and technology demonstrations launching on board SpaceX's 32nd Commercial Resupply Services mission to the International Space Station contracted by NASA. I'm Patrick O'Neill, Public Affairs and Outreach Lead for the International Space Station National Laboratory, and I'll be your moderator for today's event. First and foremost, let's talk about some logistics for this launch. As mentioned, this will be NASA's SpaceX CRS-32 mission to the orbiting laboratory. Launch is slated to take place from Launch Complex 39A at NASA's Kennedy Space Center no earlier than 4:15 a.m. Eastern Time. So, for those of us that are on Florida Space Coast, you have been warned. SpaceX's Dragon spacecraft will be filled with critical research and supplies that will be launching on a Falcon 9 rocket as part of its approximately 30-day mission to the orbiting outpost. That said, along with supplies, one of the reasons we are here is to talk about the critical component of this mission, which is launching innovative research to the space station that will be performed by the crew in the coming weeks and months. So again, that's why we're here today. What are some of the projects that are flying? Why are they flying? And what do we hope the space environment can bring to these investigations to benefit humanity, further NASA's goals of human exploration, and drive business models in this growing space economy? We have an exciting lineup of speakers showcasing the diversity of experimentation that the space station enables. These representatives will provide an overview of their project and answer questions for you, the media who have registered in advance. And we thank you, by the way, for taking the time to do so. And we also will be having the public join us via live stream. So welcome to the public. Okay, before we hear from the teams launching investigations on this mission, we will first hear from the ISS National Laboratory as well as NASA. So for media, after these remarks, I will open to any questions. To ask a question, please use the raise your hand feature within the Zoom platform, and we will unmute your line so that you may ask your question, or you may ask a question within the Q&A box. For the general public watching via live stream, if you would like to ask a question about any of the topics covered today, please use the hashtag ISS National Lab, and we will vet those accordingly. Okay, I think that puts us in a good place as far as logistics are concerned. So what do you say we start talking about space-based R&D? And to kickstart that conversation, I would like to invite Dr. Michael Roberts, Chief Scientific Officer for the ISS National Laboratory, to provide some remarks.
Thank you, Patrick. Good afternoon or good morning for those of you who may be on a different coast than I am. It's great to be here with you today ahead of SpaceX 32's Commercial Resupply Services mission for NASA to the International Space Station. These missions are critical to our ability to launch and support research performed by our on-orbit crew, and we are excited to sponsor a diverse mixture of experiments as part of this launch. We're even more excited that you'll be able to hear about these experiments from the investigators themselves. As some of you may know, our role at the ISS National Lab is to enable access to the station for the benefit of life on Earth, while also helping to foster a growing economy in low Earth orbit. The Center for the Advancement of Science in Space, or CASIS as it's known by its acronym, has now been managing the ISS National Laboratory since 2011. And during that time, we have sponsored more than 800 payloads to the space station. Working in tandem with our partners and colleagues at NASA, we seek to maximize utilization of this incredible research vehicle in space by partnering with other U.S. government agencies, industry, and academic researchers to leverage our sponsorship of up to 50% of U.S. resource allocation and crew time to the International Space Station. We, in partnership with NASA, are heavily involved in research output on the station. As I am sure my colleague Jennifer Buckley will touch on in her remarks, later this year marks a significant milestone in human exploration. As for nearly 25 years, the space station has maintained a continuous human presence in space, representing a beacon of partnership, collaboration, and innovation that will endure long after the station returns to Earth. We look forward to continuing this human achievement in the coming years, all the while sending innovative research to space that not only further enables exploration, but translates into new medical discoveries and manufacturing applications that promise to improve life here on Earth. To that end, I'll focus on the CRS-32 mission and how we work with our many partners to push the limits of research and tech dev in space. And I'd like to highlight just a few of these investigations and their benefits for us on the planet. Of course, the best you're going to hear is from our investigators who will be joining you after Jennifer and I have our comments. but I also wanted to highlight a couple of other investigations that we'll be launching this week. One of the advantages of leveraging the ISS National Lab is the ability to help validate new technologies that have advantages here on Earth. An example of one such project comes from investigators at Portland State University who will send Elvis to the space station. Not that Elvis, I'm afraid, but rather the Extant Life Volumetric Imaging System, which is an advanced microscopy system that uses holographic technology to deliver detailed three-dimensional views of cellular life. If successful, Elvis will allow scientists to study the adaptability and resilience of life under extreme conditions, further enabling our ability to detect life beyond Earth. We're also supporting an experiment from Sophie's Bionutrients, who is working with Bioserve Space Technology. Their investigation aims to develop plant-based, protein-rich alternatives to meat using microalgae. This investigation seeks to examine how microgravity affects protein yields in microalgae that acquire nutrition from organic compounds to grow. Results could provide insights into the fundamental mechanisms of microalgal cell growth, metabolism, and protein production. The last project that I'll highlight comes from our longstanding partnership with the National Science Foundation, with whom we partner to annually support release solicitations in the fields of transport phenomena. This includes gas, fluid, and particle dynamics, which are profoundly affected by the absence of gravity, and also in the fields of tissue engineering and mechanobiology. Both of these particular endeavors seek to advance fundamental knowledge as an important mission of the National Science Foundation so that they can lead to more applied investigations in the future. On the CRS-32 mission, the National Science Foundation is funding 3 projects, one of them for Menschlar Polytechnic Institute and supported by TechMasters, which aims to use microgravity to study fluid flow in protein solutions to better understand why protein clumping occurs during the manufacturing of protein-based pharmaceuticals. This remains a significant challenge in the pharmaceutical industry. In space, liquids form into a floating self-contained sphere, allowing researchers to study complex protein behaviors and motion without the walls of a container that may alter protein behavior when studied here on Earth. For those of you interested in learning more about this mission and the ISS National Laboratory, I encourage you to head to our website at issnationallab.org, and please visit our missions and launches page. There you'll find a dedicated overview of projects on this mission and further breakdowns on some of the investigations also launching on SpaceX CRS-32. Moreover, I equally encourage those interested in becoming part of this growing research community to look at our research announcements and opportunities page, as well as our outreach for workforce development and STEM education support. With that, we are all looking forward to a lovely night launch in the cool Florida night, hopefully on the 21st. Go NASA, go SpaceX, go Dragon, go science. And I return the dais back to Patrick.
Thank you very much, Dr. Michael. And yeah, to your point, we actually do already have a couple of features that are up right now that are focused on Elvis, as well as some of the NSF investigations that are slated to fly. So again, we encourage folks to go to issnationallab.org and learn a little bit more about those particular investigations. Next up, I would like to welcome Jennifer Buckley. Jennifer is the Chief Scientist for NASA's International Space Station program to provide a perspective from our nation's space agency.
Thank you, Patrick. Thanks to everyone for joining us today to hear about some of the research that we will be launching on our SpaceX 32 mission. As Mike mentioned earlier, we are coming up on the 25th anniversary of continuous crew presence on the International Space Station. To date, we have served over 5,000 investigators and conducted over 4,000 experiments. On this particular mission, we have over 30 new payloads that are enabled by this mission across ISS National Lab, NASA, and our international partners. I'd like to highlight just a couple of them that we will be conducting on the NASA and partnership side before we hear more in depth from some of our researchers today. The first one is our Smartphone Video Guidance Sensor, or SVGS2. This one's coming out of NASA's Space Technology Mission Directorate. It uses the space station's Astro-B free-flying robots to demonstrate control of small satellites using vision-based sensor developed by NASA. The visual locating software on the Astro-B robots is guided by beacons, which we install on the wall of the International Space Station. Results from this experiment could improve accuracy and reliability of guidance, navigation, and control systems for applications such as groups of small satellites, docking of crewed spacecraft, as well as operation of multiple robots on the ground working together. The next one is the Atomic Clock Ensemble in Space, also called ACES. ACES is an external instrument that will be mounted to the Columbus module, and this one is being launched by the European Space Agency, and they have collaboration with NASA's Science Mission Directorate. This one, as I mentioned in the title, is an atomic clock. According to Einstein's theory of general relativity, gravity affects the passing of time. So ACES tests theories of gravitation by creating a network of clocks, and ACES will link its own highly precise clock with the most accurate clocks on Earth and compare them to measure the flow of time. ACES looks for tiny changes in the laws of physics over time, which could hint at new discoveries about dark matter and fundamental forces of nature. Results could have applications in scientific studies of measurement, search for dark matter, fundamental physics, and also could help contribute to improving navigation for spacecraft as well as global satellites. Here on Earth, super precise clocks are essential for technologies such as GPS, internet security, financial transactions, as well as scientific research. We also are going to be flying genetic exchange and microgravity for biofilm bioremediation, or GEM-B2. This one is being sponsored by the NASA Mars Campaign Office. It's building on the prior experiment, and the B2 version aims to understand gene transfer mechanisms in bacteria. The goal of this is to inhibit core biofilm-forming genes in water resources aboard the International Space Station to maintain crew health and safety. It takes a look at biofilms, which are communities of microorganisms that aggregate and bind to a surface. Typically, they leave behind a slimy residue that can cause water or material fouling, or even could cause issues for crew health. Using donor bacteria, the crew will introduce DNA inserts called plasmids to the reciprocal or recipient bacteria that may interfere with these biofilm-forming genes and hopefully inhibit the formation of biofilm. And the last investigation I want to mention is Advanced Space Experiment Processor Industrial Crystallization Cassette. This is also called ADSEP-ICC. This is coming out of NASA's In-Space Production Applications, or INSPA, and this looks at the creation of golden nanospheres in microgravity. The investigation is using Redwire's Industrial Crystallization Cassette, ICC hardware, adding capabilities to their existing protein crystal facility on board. This is growing more golden nanospheres that have potential for applications in things such as biomedical research, cancer diagnostics and treatment, and golden nanospheres also hold promise for optical imaging, biosensing, and targeted drug delivery. I'll hand it back to Patrick.
Awesome. Well, thank you very much. A lot of great science is flying up on this mission, both from the NASA and ISS National Lab side of the house. So looking forward to what's going to be forthcoming in the not too distant future. At this point, if there are any questions from our friends in the media, please feel free to either raise your hand or put a question in the Q&A box. And if not, then we will run in and start the research side of the conversations. Silence is golden. So with that being said, let's jump on in. First research team of the day, I would like to introduce both Dr. Yupong Chen from the University of Connecticut, as well as co-founder of Eskra Biotech, and he's going to be joined by the other co-founder of Eskra Biotech, Marianne Snow, and they will talk to us about improving Janus-based nanomaterial production in microgravity.
Thanks, Patrick. My name is Marianne Snow. I'm the CEO and co-founder of Eskra Biotech. And Eskra Biotech is a preclinical nanomedicine company that provides precision delivery of mRNA, different types of gene editing, or other therapeutics to hard-to-penetrate tissues like articular cartilage, kidneys, and solid tumors. Our 4 previous space flights have allowed us to validate and refine the unique characteristics of our delivery technology, which is a Janus-based nanotechnology, a nanoparticle that we call a JBMP. New drugs often fail if they aren't tested early, and our flights have allowed us to prove that our products are not only stable, but have highlighted how our nanoparticles can encapsulate a therapeutic like mRNA, which is quite fragile, and render it room temperature stable for months. On SpaceX 32, our 5th mission, we'll be expanding our work on an osteoarthritis therapeutic and prepping for a pre-IND conversation with the FDA. And my partner and ESCRA co-founder, who is also a professor at the University of Connecticut, Dr. Yupeng Chen, is here to tell you a little more about our work.
All right. Thank you, Marianne. My name is Yupeng Chen. I'm a professor from University of Connecticut, also a scientific advisor of ESCRA Biotech. So as Mary has mentioned, that we have a focus on treating chronic disease, including osteoarthritis and solid tumors. We know those are very difficult to treat disease. For example, osteoarthritis, they are affecting 33 million Americans. However, there's no FDA-approved drug to reverse the disease. There are only injections to relieve the symptoms and the pain, but they cannot really treat disease. And solid tumor, we all know that is major health threat even today. But why? Why is that? The major reason is a lot of therapeutics or drug cannot penetrate those tissues. For example, cartilage, they have very dense tissue, and they have only a spacing of 20 nanometer. And most pharmaceutical products, especially those nano therapeutic drugs, are based on sphericals. They cannot get into the cartilage tissue. And the solid tumor, there are also some solid tumor are very dense, and which forms a barrier for any pharmaceutical to get into the solid tumor. So that's why we develop our technology. I think Patrick has one slide to show our nanoparticle we developed. All right, so this is our technology, and when they produce on Earth and in space, basically they are nano rod shaped delivery vehicles, so they can encapsulate drugs and they can target the diseased cell and the tissue because they are nano rods, but not sphericals. So they have additional advantage in penetrating those diseased tissue, including solid tumor and arthritis, arthritic cartilage. And you can see on the left, those are the nanoparticles produced on Earth. Because on Earth, we have segmentation faults, we have all sorts of disruptions. So when we produce nanoparticles, we can see some super large nanoparticles, or we can see a lot of free drugs. However, if you can see the figure on the right, sorry, on the left is the one we produce on Earth. On the right is the one we produce in space, and they form much uniform structures. And because the improvement of the uniformity, we can see that they form much smaller, those round-shaped nanotherapeutics. And those nanotherapeutics has a super good ability to penetrate those diseased tissue. In our bull and cartilage model, we have seen a 7-time increase of our nanotherapeutics to penetrate those cartilage tissue. And in the adenocarcinoma animal model, which is adenocarcinoma, as some of you may know, is a very aggressive type of cancer. In those animal models, we saw that our nanotherapeutics increase the drug accumulation into those solid tumor by 300% compared with traditional sphericals. So those space-made nanorods really improve the treatment efficacy of the current therapeutic drug. And besides producing those nanotherapeutics, we also have a parallel product line that we use our nanomaterials to engineer tissues. So we make those personalized tissue on a chip technology. For example, that we successfully produce cartilage tissue chips using patient cells so that we can use those tissue chips to as a drug screening tool. Not only will produce a pharmaceutical in space, in the future, we will also be able to test and evaluate those pharmaceutical in space. But in short, I must say that the space is such a beneficial environment for drug producers like us, and we can produce better drugs and therapeutics to help to save people's lives.
Thank you very much, folks. We appreciate this and want to say thank you to all the folks who have supported us in these efforts.
Fantastic. And real quick, if I may, this is and one of the things I love about a lot of the teams that are speaking with us today is this is not their first foray into leveraging the space station. Can you maybe describe, the fact that your team has launched multiple investigations over the last couple of years, and maybe how those projects have helped to kind of build on one another and inform you for what to expect with this launch?
So this team has taken a very streamlined approach from the very start. One of the things that's happening in drug discovery is it can be very cost prohibitive, and so we took a templatized approach and have really been looking for ways to simplify the process. Not only because the constraints of space travel, but also because we're looking at it from an economics point of view. The more that we can be cost effective in accelerating product development, then the more we can actually deliver a product to a market that is cost effective and ultimately rolls down to better price for a patient. Yupeng, do you want to talk any more about that?
Yeah, definitely. Excellent question. Actually, because space is a very different environment on Earth, even very basic lab techniques, we need to design a different system so that we can successfully run it in space. So previously for 4 missions, each time we always get a very good experience and were able to progress our science and technology along the way. And for to make any real product is from those space manufacturing environment, I think we have a long time commitment on these goals. And we build our experience and by each missions, and eventually it will lead us to our final goal, which will produce a drug to help patients on Earth.
I think the other thing too, Patrick, is Yupeng and I have really had the privilege of tying into an ecosystem that is working hard to build a sustainable economy in low Earth orbit for products like this. And so by utilizing the resources that are available to us, building network connections, and also really following a lot of methodologies that you would follow in commercial drug development, but doing it in this unique environment and leveraging the unique environment to accelerate discovery has been a real benefit to us.
And one thing, so I had the opportunity to meet with both Yupeng and Marianne a couple of weeks ago when we were filming a video, which should be going live tomorrow, as a matter of fact, in association with this investigation. So encourage everyone to check that out. But one of the things I thought was pretty fascinating across the board is not only is it the accelerated opportunity for a drug discovery, but maybe more importantly, to accelerate drug candidates that aren't going to move forward, and how that allows for you then to, so it's not just finding the right ones, but it's also finding the wrong ones and then moving away from that so that you can focus your efforts on the right candidates. Is that fairly accurate?
Exactly.
Yeah, I was going to say, just the fact that we have been given the privilege of being on station 4 times already. This is our 5th flight in 2 years. So, and I know many of our esteemed colleagues have been at this a very long time. And they've got a lot of missions to their credit, but we're moving towards a more commercial cadence, and I think that that's something that's really important. But and I think that this is particularly important because optimization is always part of the drug development process. But I think doing it in a commercial context not just basic academic research, but our end goal is to bring a product to market. Sorry, Yupeng.
No problem. I just want to add a few things from a technical aspect, because we are not just the drug producer, we are also creating those disease models, as we mentioned, those tissue on chips. And because we are focusing on chronic disease, arthritis, solid tumors, it takes a long time for them to develop on Earth. But in space, it definitely have accelerated disease progression and can accelerate our drug testing, and so that we can screen and identify the most effective drug candidate much better. And because we produce drugs, we have a better sense about what type of disease models can work better. So we use those patient cells to create those personalized models. And we talked with FDA expert about our models, and potentially it can also reduce the use of animals, and can also help the can also accelerate the FDA review process for the approval of our drug.
Fascinating stuff. It's been a pleasure to work with your team. And again, I understand that this is not going to be your last foray into space, and we look forward to continued success that you all have. And again, best of luck with this mission.
Thanks so much, Patrick, and
Thank you.
Godspeed to all of our colleagues who are putting stuff on.
Fantastic. Okay, well, now we are going to move on to Dr. Claire Fortenberry. She is the research aerospace engineer from NASA's Glenn Research Center in Cleveland, and she will be highlighting the monitoring of aerosol pollutants in space.
All right. Thanks so much, Patrick. Thanks for the introduction. So as Patrick said, my name is Claire Fortenberry. I'm a researcher and a particulate monitoring specialist at NASA Glenn Research Center. I am also the PI for the Exploration Aerosol Monitors flight demonstration from the Life Support Systems project. This was funded by the Mars Campaign Office at NASA. So lofted cabin dust, we know from all of the years that we've been, that we've had a habitated crew in space. We know that lofted dust is a nuisance. We know that because there's no gravity, lofted dust doesn't settle like it would on Earth. So we have to have filters to remove it. And Patrick, if you could please pull up the pictures that I've provided. Thanks very much. So you can see in the bottom right, that is a picture of accumulated dust on the ISS's bacterial filter element, or BFE. And this is after about 8 days of continuous use. So the crew has to go and vacuum the dust layer off of the face of the filter because it accumulates so rapidly. So it can be a nuisance. It can cause false smoke alarms, and it's generally there's a need to monitor and control for it, even now with the ISS. Cabin dust on station, it's composed of a variety of different types of particles from different sources. In the top right, I'm showing some micrographs from a previous investigation of ISS dust. It comes from a variety of sources, including material wear and tear, so stainless steel and fiberglass, iron, for example, skin flakes, clothing fibers, and then this antiperspirant particle we were able to identify as such because of certain chemical inclusions. And then there's some particles that we still are not entirely sure exactly where they come from, like the silver dendrite particle. We're still not sure what is causing the formation of that. Currently, there is no system on station to regularly and continuously monitor dust. But as we continue to prepare for increasingly Earth-independent operations and missions, like our mission to the moon with Artemis, and then future missions to Mars, the need for a continuous monitor is going to grow to inform the crew about their environment over time. One of the examples that we're thinking about now as we're returning to the moon with Artemis is the potential for lunar dust intrusion in particular. Lunar dust is specifically of concern because particles are pretty sharp, they can be quite small, and they can be small enough to deposit deep into the respiratory system and potentially cause harm to crew health, especially if there's prolonged exposure. So we are sending the Exploration Aerosol Monitor Suite to station to demonstrate 3 different aerosol monitoring technologies, each of which provides a complementary and important piece of the airborne particulate environment puzzle on station. So there are 3 monitors that I'm showing in the picture to the left. From left to right, we have the optoelectrical real-time aerosol classifier, or the OPRA, the moderated aerosol growth with internal water cycling monitor, or the MAGIC, and on the bottom there is the mobile aerosol sampler, or the MASS. And these 3 monitors, each of these were developed by a small business through a NASA SBIR or an STTR award, and this payload was a result of their hard work, as well as the hard work of Bioserve Space Technologies, who are our integrators, our implementation partner for this payload. So this work is building off of a previous flight demonstration called the Airborne Particulate Monitor, or APM, that is still currently on station. It is sampling in the FGB. We are trying to close remaining capability gaps with particulate monitoring in advance of longer duration exploration missions. And these capability gaps include reducing mass, power, and volume. We want to be monitoring very specific aerosol particulate parameters, a parameter called mass concentration with high accuracy. And we also want to be ultimately moving towards the ability to distinguish between different aerosol classes. I mentioned previously that that dust can cause false alarms due to false smoke alarms due to nuisance dust. The OPERA features the capability to reduce false alarms by distinguishing between different classes of aerosols. In addition to our active exploration aerosol monitoring suite, we will be deploying passive aerosol samplers, which have been flown before. These were used to capture the micrographs that I'm showing to the right in a previous investigation. This will be used to continue to investigate chemical composition, morphologies, ultimately sources of airborne particles on station. So when we do this investigation, we will be taking these monitors and moving them to different areas on station to monitor for a continued period of time, about a month at a time, for 7 locations, for a duration in total of approximately 7 months. And we're targeting different areas and different activities of interest that some of which we know to be dust generating based on previous research, and some of which we have not investigated fully yet, including a vehicle docking undocking situation. So we are hoping to get new data to complement and to build upon our previous work, with the ultimate goal of, again, flight proving these technologies for potential infusion of this technology into a future exploration program. So that's what I have, and happy to take any questions.
Awesome. Well, thank you very much, Claire. I see that we have a hand up from Will Robinson Smith.
Yeah, thanks, Patrick, and appreciate you taking the time to talk with us today. Going to the point you made about working in this technology at future long duration missions, have you started having conversations with some of the HLS providers that are currently onboarded with SpaceX and Blue Origin about how to integrate this into their particular landers, or how this might weave into the Orion spacecraft or Gateway? Thanks.
Yeah, conversations are happening. That is a bit above my, I think, ability to answer at this time.
But that's why we hope for the best with this investigation, right? That it sets the foundation for future conversations and integration of next steps. And I guess one of the questions I was going to have, Claire, was, and I think that you did kind of touch on it, but you had like the 3 sensors, the 3 monitors. Is the thought, let's validate this right now, but is there an idea for us to maybe turn that into like a new system that would eventually be used either on this space station or future space platforms, or going beyond low Earth orbit?
Yeah, I mean, ultimately, the goal of any tech development is to, we talk about closing capability gaps. The gap is really never truly, in my view, you're never really going to close. You're always going to make progress towards something better. You're always wanting to progress towards something better. So in this case, the way that I see that is you have these 3 different monitors, each of which has unique capabilities. And I think, a good example of that is you have one monitor that returns, one property where you are able to potentially measure a size range that the other might not be able to capture, but that other is able to, for example, capture a different property that is needed to verify another NASA requirement. So I think ultimately, yeah, if it's a balance between, the I guess the fidelity of your measurement and meeting the requirements of NASA and the providers, and the mass power volume. I think that's the mass power volume is always going to be a challenge. So certainly in a perfect world, you'd have all 3 and you'd have the perfect picture, of the environment, and that is what we would want to be striving towards. So that's how I envision, the future of this work.
Well, again, this builds on previous research, so hopefully this leads to future investigations, whether that be on station or beyond. So best of luck to you and to your team, and go forth and conquer.
Yeah, thank you, and good luck to all of the other investigators as well.
Thank you very much, Claire. Next up, we're going to move on to Major Travis Tubbs with the United States Air Force Academy and Dr. Heath Mills of Rhodium Scientific. And they're going to be talking about a series of investigations actually looking at plant and route changes and development in space. So with that, Travis and Heath, I'll leave it to you all.
Perfect. Appreciate it. Thank you very much for the time. So I guess let me introduce myself real quick. My name is Major Travis Tubbs. I am considered still a biology professor at the Air Force Academy in Colorado Springs, but I have also been detailed out to Chile. So I'm calling in from Chile down now, and I work for the Air Force Office of Scientific Research as an international program officer. So my job is to go out and find and fund the best science that I can. But in my previous hat as a biology professor, my job was to investigate the biological systems and understand how they worked. I'm also a Space Force officer. And so being in the Space Force automatically made me interested in space. And so that's how we got involved in this whole process to begin with. And currently, we are looking at launching some of our experiments into space with the aid of Rhodium. Rhodium has been a great partner with us. We've used them in the past for a previous experiment on the International Space Station, and that's where Heath comes in. Heath, if you want, go ahead and introduce yourself, and then we can maybe talk about our current experiment.
Thanks, Travis. It's been great working with you, Travis, and especially also with the ISS National Lab and NASA. My name is Heath Mills. I am the Chief Scientific Officer for Rhodium Scientific. Rhodium is the commercial service provider implementation partner for the work that Travis will be talking about today. We are one of the, we're the first biotech space company, and working with this team to really develop the science aspects that you'll hear about from Travis was very important to us. We really look at seeing ways to advance that science output, to understand and really make new discoveries that will help life back on Earth. So with that brief introduction, I'm sure we'll talk a little bit more, but Travis, I'll put it back to you, and looking forward to hearing about the science.
Perfect. Thanks, Heath. All right, so just real quick, what this experiment is, this is called the Rhodium USAFA NIGHT program, and NIGHT stands for non-photosynthetic inquiry into growth habitat traits. What that means is we're trying to understand how do we grow plants more efficiently, more effectively, and hopefully, as time goes on, we can learn how to bypass photosynthesis. That's kind of an exciting possibility, but this is one step in that process. So to do that with this experiment, this is an experiment where we use what's called space tomatoes. So space tomatoes stands for small plants for agriculture in confined environments. And it works really well because they're going into space. So these space tomatoes are recently developed. They're gene-edited tomato plants that are extremely dwarf and ideally suited for growth in space. This experiment aims to enhance our knowledge of plant growth in space, as well as investigating how the genetic modifications and heterotrophic growth influence plant development, as well as the gene expressions. So this is a follow-on experiment to what we did in the past. The United States Air Force Academy's astrobiology program, our space biology program, they help to establish a scientific baseline for how we enhance biological materials using the space environment. The goal here is to understand how can we bypass photosynthesis. And if we can do that, if we can make, we've shown on the ground that we can feed acetate and we can feed these plants through their root system in a way that will allow them to grow without using photosynthesis. So this experiment is a way to test that. And it helps build on our previous experiment where we were using Arabidopsis, which is the model organism for plant growth. So this is an overarching goal that we want to do. The ultimate goal is to develop a mechanism for biomanufacturing these polysaccharides using the waste carbon dioxide and the light that is around to produce to reduce the logistical costs of sending things into space. We can use this waste reutilization mechanism. So the life science technologies may help provide a solution for space challenges, but also could help here on Earth, understanding how do we grow plants more efficiently, more effectively, and reduce some of the costs as well as the time that's required growing these plants. This is a follow-on of what we've already done in the past. And if you don't mind, Patrick, go ahead and pull up our slides. It'll show a little bit of what we do. So first thing we want to understand in our first experiment was understanding how gravity affected plant growth. And so this first one talks about gravitropism. What is it? So one of the things that we did in our original experiment was understanding how do plants grow in space. And this has been done in different ways out in space. But if you look at our first picture, you can see that there are these dark nodules. I like to call them little marbles. They're like little marbles that are in the tips of these roots. These help give direction to plants in the world naturally. We also had a genetically modified one where those little marbles, they're called statoliths. These marbles have been removed. So we wanted to compare how do these plants grow differently if you take away their means of assessing gravity. How do they grow differently? And that's what the results here. These results just came back from the previous Crew 9 mission, but the results are here where we can actually see some of the wild type roots where they grew. On the bottom line, you see that they grew very straight and very consistently. The mutants, you can see their roots were kind of all over the place because they couldn't sense gravity. What we wanted to see is if you put these in space, do they start to mirror each other? Do they start to look similar? Do the wild type plants who should know where gravity is, do they behave differently now that we've taken the standard Earth gravity system away and they use these microgravity? Do they start to appear that way? And that's what we're investigating and looking like. But initial inquiry starts to look like, no, once they kind of get started on a path, they kind of tend to stay on that path. If you go to the next slide, please. This is the pictures of some of the things that we've done. So the first one is a cuvette that we launched, and these cuvettes are kind of there's our little, if you will, laboratory that we send it into space and we grow the plants. You can see the root structures. We're going to do the same thing with this launch that's happening here. We're going to try and understand how these roots develop with these space tomatoes. But we take the images and then we can actually 3D model what the root structures are. And here's a picture of our little Sophia is what it's called. It's our space optimized plant habitat in array. This is our laboratory where we grow these plants, and it's a very small, very efficient, very unique little structure that allows for any variety of science that we can fit into it. And we're looking at plants at the moment. This has been done before. We launched it on the Polaris Dawn mission, which is a unique one. It's not with the ISS. We launched it before we launched on the ISS. We we did it on the Polaris Dawn mission. And if you look here, you'll see Jarek Isaacman. He is the current nominee to be the NASA administrator. He is there in this picture because he was the crew commander of the Polaris Dawn mission. And they're holding up our little Sophia box. So bottom line is this night mission is a great opportunity for us to test how do we grow food better in space? How do we grow it more efficiently? How do we make it something that not only helps the astronauts and the future of humanity as we move off of this planet, but how do we help ourselves here on this planet now? How do we help ourselves to grow food better? How do we understand the basic mechanisms of photosynthesis, and what can we do to make that more cost effective and more efficient in production of food? And Heath, is there anything you'd like to add?
One quick note, the USAFA side on this, U.S. Air Force Academy has been a great partner with Rhodium to develop the flight hardware that you see there. This flight hardware was designed with support from several cadets. They were able to visualize this concept to be able to do these arrays of plant growth chambers. And we were able to work with them to get this flight certified. It's flown on now 2 vehicles so far, and we're about to go to the ISS a second time. Not only can it grow plants, but it's also robust enough to grow fungi in an upcoming mission, as well as bacteria and other micro eukaryotes. So great partnership with USAFA, with the cadets there, and showing what they can do to put things, even though they're Air Force, outside the atmosphere and do some amazing research. So thanks, Travis, and thanks USAFA for this support.
Perfect. Thank you, Heath. And one thing that Heath, I guess we should point out is this is also a very, very cost effective compared to other systems for growing plants. This is literally just, it's 3D printed in such a way with the unique material. So it's actually been exposed to the outside of space when Jarek Isaacman's team did a spacewalk, but it was also very, very cost effective. So this is a very innovative way that USAFA has, the United States Air Force Academy has pushed the boundaries of what is possible out in space. And Patrick, if you don't mind, we'll turn the time over to any questions.
I see that there's a question from Will Robinson Smith once again.
Wonderful.
Hey, thanks, Patrick, for allowing me to take another question here. And just want to pick up on that last point that you were making. When the Dragon vehicle was brought down to vacuum, what were the impacts that you saw on the tomato growths? Were there any particular implications from being in that environment that may help foster further research? Thanks.
So you're talking about when they were exposed to the vacuum of space, and then when they were, I guess, brought back into the natural environment on the Polaris Dawn mission. Is that correct?
Correct. Yeah.
So first thing is these ones were not tomatoes. The tomatoes are the ones we're doing currently. These ones were Arabidopsis, but the impact was very minimal in that they were in their own. So the box and the environment itself, the experiment was in the vacuum of space. But as you can imagine, plants don't do real well in the vacuum of space. So they had to be contained inside of these little cuvettes in their own natural environment. The nice thing about plants is they can survive in their own environment because they produce oxygen as well as carbon dioxide. So they could stay there until the food runs out. But they never were exposed to the vacuum of space because the plants would have died almost immediately. But because they were inside these little cuvettes that were sealed, they never had to experience that vacuum. But the box and the structure itself did experience that vacuum. Does that answer your question?
Yes, thank you.
Perfect. Any other questions?
Gee, I was going to have a quick question on my own, and it equally kind of builds on the differentiation, if you will, between launching an investigation for the Polaris Dawn mission, which obviously is not at the same trajectory as this space station is. So you'll have 2 tries at sending investigations to station, but maybe can you describe kind of the differentiation of how you prepare a project between station and the Polaris Dawn mission?
Yeah.
Well, I would like to say it is almost night and day. It is about as different as I could experience. And I think one of the reasons why that's so, for one, it was our very first attempt using Polaris Dawn, and SpaceX was an amazing team, but they were also doing the very first, this Polaris Dawn was the very first commercial launch out in with a commercial launch with a spacewalk associated with it. So they had to be super particular about what was going to be on there with the science and make sure everything was secure and safe. We found life to be much, much easier once we found rhodium, because rhodium was much clearer on how to do things the NASA way, to make sure that we did it the way that made sense on the International Space Station. And thanks to their help, our life got much, much easier, because we were very much trying to muddle our way through on the Polaris Dawn mission. SpaceX did a phenomenal job, but with rhodium helping us and with NASA helping us in this, the second launch, it went much smoother, much quicker, and much more efficiently for us at the Air Force Academy. And we learned a lot of lessons each step of the way. I would say this one has been almost as streamlined as it can be for this launch, thanks to rhodium and thanks to the partners that we're working with. We're working with the University of California, Riverside, and they're doing an amazing research on the science piece of these plants. And USAFA has paved the way for us to make this a continually process to launch experiments into space. And we hope to continue to do this process as long as we're allowed to.
We appreciate that, Travis. And I just want to make sure it was clear that you said that life was better with rhodium. So I appreciate that. Thank you.
Technically, that is probably the right way to say it. That is very true.
The ISS National Lab neither endorses or does not endorse that last comment from Dr. Mills. Thank you very much. Well, again, congratulations on the first two launch attempts, and hopefully this one continues to go out there and build on the knowledge that's being gained. So again, we'll be on the lookout. So thank you very much.
Wish us all the best of luck.
All the best. All right, now last but certainly not least is going to be Dr. Sarah Wyatt from Ohio University, who will discuss the NASA-sponsored experiment assessing the effects of space radiation on the genome and telomere of a plant that I'm always mess up its name, so I'm not going to try. So instead, I'm just going to let Dr. Wyatt provide some details on her investigation.
Okay, so this investigation actually started with the twin study that happened several years ago now. One of the things that Dr. Susan Bailey and her lab looked at were telomeres of DNA. Telomeres are the natural ends of the chromosomes, and they shorten with cell division and with a variety of different environmental stresses. And in space, what they saw with a twin that was in space was the telomeres were much longer. So the telomeres were longer, but then when he returned to Earth, they shortened back down really quickly, which really isn't a good thing. So telomeres are a molecular determinant of aging and aging trajectory. So what we wanted to do, when I say we, I mean Dr. Shippen's lab at Texas A&M and my lab at Ohio University, was look at how telomeres responded to spaceflight in plants. So this is an Arabidopsis. That's a really tiny plant. If you look at the bottom right of the slide, you see the little Arabidopsis seedlings growing on a petri dish. This is how we flew them. So we have about 15 across, and they're flown seated across, and then 4 rows of them. And we'll get back to this in a minute. But so we collected these and looked at telomere length. And what we discovered in the plants was the telomere length really didn't change much, which was kind of surprising since we saw such a marked response in humans. But we also had the opportunity, because we had more material and more capability to do things with plants that you really can't experiment on with humans. What we saw was that telomerase, the enzyme that creates the telomeres, actually was significantly increased in plants. So, and I'm talking about 150-fold increased in plants. However, remember, the telomeres aren't any longer. So then our question became, what is the telomerase doing? So this led to a new proposal and this experiment, which we euphemistically refer to as RAD plants, radiation plants. So in this experiment, we're actually flying 6 different genotypes. I won't go through them all, but there are enzymes. They're either plants that are overexpressing certain enzymes or knocked out in those enzymes that are important for telomeres and radiation response. So we've got 6 different plants, and we're going to be looking at their responses in flight, both telomere length and telomerase in each one of these to try to determine what the mechanisms are that are occurring in flight. So this will be done in 3 different locations. So we have the simulated galactic cosmic rays at the National Lab at Brookhaven, which is the top left image for you. We also have low dose radiation, neutron and gamma radiation, a prolonged exposure. So this was weeks of exposure. And this is at Colorado State University, where Dr. Bailey works. And then we have the space flight itself, which we hope is going to launch on Monday. We're headed to Kennedy tomorrow to prep for that flight. And what you see on the lower set is the 2 students and postdocs that are working on this, Borja Barcelona and Emma Canaday from OU. So this will be 5-day-old seedlings. They'll be launched as seeds. They will germinate and grow for 5 days, and then they will be frozen and brought back to Earth. Once they're brought back, we will be looking at various markers of growth, but also markers of radiation effects. So looking at telomere length, other DNA damage, and telomerase activity in each of these mutants to see if we've got any kind of effect on telomeres in general or on telomerase activity, so we can try to dissect the molecular mechanisms that are occurring here. We also have a variety of other genes that we can look at through RT-PCR, real-time PCR, to see if they're going up or down in any of these plants, to see if we can further expand on our information in these processes. So hopefully in the next month or so, we'll have some really significant results on what telomeres and telomerase is doing in spaceflight and how that may be either being affected by the radiation levels, or we can also, through this experiment, sort out any effects of the microgravity versus the radiation. So we're hoping to have some really good information on this that we can apply generally or test generally across all species. And with that, I will give it back to Patrick.
So you actually answer kind of towards the very last portion right there. I was going to follow up with that and see, obviously this is focused on plant biology research, but you also alluded to the twin study and the telomeres. But, based on this investigation, are there things that we can glean that could be helpful for us with human health, either in space or here on the ground on Earth?
Indeed, one of the things that's really great about plants, so we can look at telomeres. Telomeres exist in all organisms. So what's happening in plants may also be happening in humans. So to see if some of these different enzymes that we're checking, so we're looking at catalase, that's one of the enzymes we're looking at, which will mitigate reactive oxygen species, which are generated during radiation. If we see various increases and decreases, that's something that we can take back to animals in general and look to see, is this a problem? And then how do we deal with it? So we not only learn how to better grow plants, so for long-term missions, when we're going to have tremendous radiation, depending on where we are, if we're still on ISS or if we're going further out to Mars, then we're going to have potentially mediations for this radiation damage, not only that might apply to plants, but that we can test in animal systems and in humans to offer protection.
Another thought then that popped into my mind too is, so you just said that, using space to better grow plants, but I also have to imagine too that using the space station at this juncture, we have a far better understanding of how to protect the plants that we are trying to grow.
Indeed, we do. So we have a much better idea of, the whole idea of not just kind of space plants, but space crops. So we just talked about tomatoes. If we're seeing these kinds of effects in tomatoes, especially if they're multi-generational, then you're going to start seeing plants not be able to survive multi-generationally. And if that's true, we're going to lose, there will be no farming if we can only do it for a couple of generations and we have to start over again. So trying to make sure that we have multi-generational ability to grow food and we use plants for everything. Who are we kidding? Your clothes you're wearing, everything we're doing. So to have some idea that we could take all of this with us to explore is a little naive, but we could take plant seeds, various seeds to different plants, widely to almost anywhere. And if we can figure out how to grow them in these different locations or how to grow them at different times, then we have the supplies that we need.
I have seen the movie Interstellar and seeing some of the issues that they were having in trying to grow plants out there and the issues that that would ultimately create. So I certainly agree with you there, where it's kind of the building blocks of life.
Yes, it is.
Awesome. For that, I'm going to put out and see if there's any questions from our friends in the media. If not, then we're going to get ready to wrap things up for the day. But thank you very much, Dr. White. That was great.
Thank you.
Okay, one more second. Okay, well then with that, again, this has been a lot of fun. Hopefully it's been informative, not only for the members of the media that joined, thank you for that, but also for the general public that had the opportunity to watch via the live stream. So again, at this point, I think we're going to move on and get ready to call it a day. So as mentioned, we are looking at launch no earlier than Monday morning at 4:15 a.m., which would be April 21st from NASA's Kennedy Space Center, Launch Complex 39A. For up-to-the-minute information on that launch, please follow at NASA, at space_station, at NASA Kennedy via social platforms, or follow the coverage on NASA's International Space Station blog. And again, if you want to follow on the ISS National Lab, we'll be having a variety of content that will be being showcased on our website in the coming days about some of the projects that were described today, as well as other projects that were not on this webinar. But with that, again, thank you so much. Really appreciate you all being here today. On behalf of the ISS National Lab, I'm Patrick O'Neill. Go Dragon and go Science. Have a good afternoon.